All right, guys, when we get into chapter 21 here, it seems like a very odd order to do some things in this unit. We covered chapters 13, 14, and 15, and then we jumped to chapter 21. Weird, right? There is a reason for it, and I hope that you understand that reason as we get deeper into some of the information that we are going to touch on today. Chapter 21 is entitled The Genetic Basis of Development, and the reason I've chosen to put chapter 21 here is because what it actually gets down to is what happens, sorry, I'm kind of going out of order there, is what happens when the zygote gets fertilized, okay? Or the egg gets fertilized to create a zygote. We know that eventually it develops into what we are as humans, or whatever other organism happens to have a developing zygote. And we know that we express those genes, and we know that we are what we are right now, right? And we have all these features and phenotypes and all this type of stuff. And this chapter helps us go from point A to point B. So the point at which that egg is fertilized and makes a zygote, and then point B, where we are now, there's a lot that happens in between there. And a lot of that is the expression of genes and the creation of cell types and body parts and then organ systems, all this stuff, right? And so that's what this chapter starts to get into, is what happens between fertilization and where we are now. And it's a very, very complicated picture, and this chapter looks to at least touch on it. Okay, and that's about all it does, and even just touching on it is very, very difficult to comprehend and understand sometimes. So we're going to try our best and use this picture here as motivation. And I will have talked about this picture in person before you see this video, but effectively what you see here is a fruit fly that has two eyes. One of them is normal, the other one is not. This guy right here is not a normal eye. It is an eye that actually is developing from the tip of his antenna. And so your motivation, I guess, in terms of this chapter, since some of it can be kind of confusing at times, is to gain at least some understanding of how this would have happened. Why on earth is there an eyeball sticking out of the antenna of that fruit fly instead of where it should be, like this one is right next to it? Okay, that's some of the stuff that we're going to get into in this particular chapter. Okay, we're going to start at the beginning. We're going to talk about how embryonic development involves cell division, cell differentiation, and morphogenesis. So I mentioned before that a zygote can give rise ultimately to what we know to be ourselves, right? That we are walking, talking humans with a certain type of eye color or hair color or skin color, whatever it might be. And each one of those things that I just mentioned is governed by our genes, right? So, we ultimately are a function of all of these different cells which are coded for by our genes. And each cell that make us up can have a different structure, and as a result, have a corresponding function. Now, the development of that zygote from the time it's fertilized to where we are now involves three processes: cell division, morphogenesis, which is also known as the creation of form, okay, and then cell differentiation, which is where cells become specialized in their structure and function. So we can kind of look at this diagram. It shows this shows the development of two different types of organisms: a plant at the bottom and an animal at the top. Okay, we've got a starfish at the top and a plant at the bottom. Okay, and so the development of these organisms takes place obviously over the course of time, and we need certain things to happen with the early zygote to get it into a form that we know and love, right? And so what we have to first do as a little baby zygote is we have to divide, right? We are trillions and trillions of cells. A human being has trillions and trillions of cells. The only way to get to trillions and trillions of cells is if we divide that very first one. So one of the first things that has to happen is we have to amass a bunch of cells. And that's what we see happening here. We amass a bunch of cells through cell division. Then, once we kind of get enough cells and we get enough bulk material to do something with, we start to create our body form. And we can kind of see that here at this 8-cell stage and kind of in between there, all the way up to 32, 64 cells. We're just kind of a solid ball of cells. And at some point, our body starts to have a form. Okay? Morphogenesis starts to occur. And we notice that this solid 8-cell ball, which kind of further divides, ultimately develops into what we call a blastula. And this is one of the earliest forms that we have, just kind of now it's a hollow ball of cells. It's a sphere that's hollow. And as we work our way through the life of the animal, we see that we have more form creating here in something known as gastrulation. These are not terms that you necessarily have to remember for a test or anything, but what this is creation of an early gut. Okay, and so we start to see at this point observable cell differentiation, where now some of these kind of generic cells that we saw in the blastula and the 8-cell stage, they start to get jobs. And that's really what makes us us, right? I've got cells in my eyes that have specific jobs. I've got cells in my heart that have a specific job. I've got cells in my liver that have a specific job, and we're thankful that they do, because it takes care of all these different scenarios that our body finds itself in. And so you notice that each one of these things continues throughout the edge. My body is still dividing. It's still, the cells are still dividing. My body still at some point does create form, okay, when it needs to. You guys at one point have seen this nasty fingernail, right? Well, it still looks like a fingernail, which means it's creating form as it grows back out, right? And cell differentiation still occurs. It has to, right? If I wreck a heart cell, I would want some of those heart cells to grow back and have that differentiated heart cell look to them, okay? So those are some of the things that we're going to start to talk about in this lecture, okay? So, all cells, one thing you have to understand as we embark on this little journey of deciding what happens from zygote to adult, is that we have to understand that all cells in our body have the exact same DNA and thus the same genes. They call this genomic equivalence. So this begs the question, why and how do we have different types of cells? Now, it's not one of those things you oftentimes think about, but the cells at the tips of my fingers have DNA in it, right? It has all 46 of my chromosomes in them. Okay? All 46 of my chromosomes are in the cell at the very tip of this finger. And you notice that it looks like a finger. It doesn't look like anything else. It doesn't look like a head. It doesn't look like a eyeball. It doesn't look like anything. It looks like the tip of a finger, which is what we should know that it looks like. But these differences that we see in different cells, so the differences that I see in the tips of my fingers compared to my eyeballs, compared to my heart or whatever, are due to gene expression, okay? And these differences arise during development, different stages of development, as different regulatory mechanisms in our DNA will turn certain genes on and off. So we have to ask ourselves, first off, what does it mean to express a gene, aside from the poetry and the rhymes and the song making that I just did? What does it mean to express a gene is a question that I've probably already asked you. So you probably already know the answer. It means to turn on a gene. It means to take some gene that's in your DNA and make something from it, okay? So to express a gene means nothing more than to turn a gene on, okay? Now let's extend it a little bit further, because it'll ultimately lead to the point that we're trying to make here. What does differential gene expression mean? And again, you probably have already been asked this question and maybe struggled through it. But it means that different genes are expressed in different cells. Differential gene expression means that different genes are expressed in different cells, and if you want to go flip that, you could say that certain genes aren't expressed in certain cells. So, for instance, at the tips of my fingers, the tip of the finger cells are expressed. Okay? You know what ones aren't expressed? Eyeball genes. Okay, that would be creepy, right? Seeing eyeballs budding out of the tips of my fingers. But I can guarantee you the genes that code for eyeballs are still in the cells in the tips of my fingers. Those genes are just off. The genes that are on are the ones that code for the tips of my fingers, right? Just imagine what it would look like if you had eyeballs coming out of the tips of your fingers. Well, I do want you to also understand that science can kind of do that. Okay, we're on the verge of being able to possibly, if you really wanted to, grow eyeballs out of the tips of your fingers. But science does have the ability to reverse the off switch on some of these genes. It can take the genes that were previously off and then turn them on, or allow them to be turned on. Okay, so what may be involved in cell determination and differentiation? What creates this ability for the tips of my fingers to look like tips of fingers, or my eyes to look like eyes? What does that? Okay, that's what we're going to start to get into in this section here. Okay, and it really kind of boils down to, I'm lying to you a little bit, it boils down to probably a lot more than these three things here, but these three things are relatively simple to understand, and they're things that we can illustrate here. And they do play a role in determining what your early generic cells are going to do, and also then giving them that job. Okay? So transcriptional regulation, cytoplasmic determinants, and induction of embryonic cells all play a role in the determination and thus differentiation of your cells, okay, in that early development. So we're going to talk about each one of these phenomenon a little bit more in detail as we work our way through. Okay, so the first one we'll discuss a little bit more in depth is something known as the transcriptional regulation of gene expression during development. It's a lot of big words put together, and I want to kind of break that down a little bit. Okay. Transcriptional regulation means that we are regulating the process called transcription. Okay, let me just overview or review what that means. I'm trying to get a thing where I can write. Okay, so when we are transcribing a gene, we are taking DNA and we are turning it into mRNA. Okay? Effectively, what we have to do here is turn that chunk of DNA on. Okay? That chunk of DNA is also called a gene. All right? And so when we want to turn this DNA on and make mRNA, we have to transcribe it. All right? It has to go through, this gene in the DNA has to go through transcription. And transcription is actually a process that can be regulated. It can be turned on or off, right? And so that's what we're going to talk about here in this particular part of the videos. We're going to talk about how it is that we kind of turn these genes on to then make the mRNA that ultimately makes the protein that is us. We are the protein. Okay, we are a conglomeration of many, many different proteins all working in concert. Okay, so that's what we're going to start to get into and where we're going to be. Okay, so we have to talk about this in two different levels. We have to talk about this, number one, in the vein that we have to understand that early on, our early developing embryo, which is our early body, is a very generic ball of cells, okay? And certain cells have to get jobs. Certain cells have to determine what they want to do, okay? So cell determination precedes differentiation, and it involves the expression of genes for certain proteins, okay? And then these proteins will enable differentiated cells to carry out specific tasks. And I think when we actually start to see all of these things in one diagram, it'll make a little bit more sense. And I'll throw a couple of analogies at you that I think maybe will help us out as well. Okay, so let me go ahead and snapshot this thing, because I think I'm going to try to draw all over this. All right, so what we have to understand is that this embryonic, let me change the color here. Let's go with red. This embryonic precursor cell is generic, which means it doesn't really do anything. Okay, it lives, it's a cell that lives, it has no specific job. It doesn't necessarily mean that it won't have a job, but at its early stage of development, the embryonic precursor cell does not have a job. It's very generic. Okay, and what we have to understand about the generic cell is that at this point, so early on in its development, almost all of the genes are going to be off. Okay, and if we want that cell to have a specific job or do something, we effectively have to turn those genes on. Okay, now this is how we regulate transcription, by turning the genes on, by providing something known as transcription factors. Transcription factors are little proteins that are created that will ultimately turn on some gene in your DNA. Okay, and I think I've mentioned transcription factors before, and here we see them again, and we're going to see them again in the next unit. Transcription factors are very important things. They turn genes on. Okay, so early on here we notice that all of these genes of interest up here that they highlight are at this point off. Okay, this one they call the master control gene, MyoD, is off. Other muscle-specific protein genes are off. Okay, and so what we have to do is we ultimately have to give this generic cell a job. Okay, it's very similar to what you guys go through in your search for college and careers and things of that nature. You think to yourself, oh man, I like this science stuff, I like this biology stuff, I want to be a doctor. Okay, so at this point you have determined what you want to do. And a cell is very similar. Cells determine what they want to do. So you notice that this is called an embryonic precursor cell up here. It's very generic and boring. And down here, whoa, it's got a new name. This is effectively like you declaring a major for college. This thing has declared a major for its life. It's decided, I want to be a muscle cell. So it has been determined that this thing is going to be a muscle cell. We'll talk about how that determination happens later. But this cell right here that was generic has been determined, hey, you're going to be a muscle cell, same way that you determined, hey, I want to major in being a doctor or pre-med or whatever it is you guys major in these days. Okay? So once this cell is determined, it has to go through the necessary measures to become a muscle cell. The same way that you have to go through the necessary measures to become a doctor. And for you, it's going to be very, very difficult, right? It's a hard road. Not everybody makes it. But you take your classes, you do your studying, you go to the lab, you do this, that, the other thing. And if you do all those things, you become a doctor. The same way this guy here, the myoblast cell, has decided, I'm going to become a muscle cell, not a doctor, a muscle cell, so I've got to go through the necessary measures. So what happens is this gene right here looks like it gets turned on. Okay, that gene creates mRNA. That gene creates a protein. It's called a MyoD protein, and it happens to be a transcription factor. Okay, and so what that does, that transcription factor that I just circled in this middle paragraph here, this middle picture, is actually going to turn on. It's going to be the on switch for this gene and also for this gene. Okay? And so these other genes that we see here, this pink one and this orange one and this yellow one up here, those are genes that code for specific things that you would find only in a muscle cell. Okay? And so what this early on transcription factor does, that I've kind of circled down here again, is it turns all these muscle-specific genes on so that you can make muscle parts. This guy's trying to make proteins and things like that he can put into his cell, right here, that he can put into his cell body and ultimately become a muscle cell. That's what his ultimate goal is, right? And so we see that flow down here. Now, at this point, he's gone through all the necessary measures. He's starting to turn on all the necessary genes. He is literally going to transform himself into a muscle cell. Okay? And so that's what you guys would do. By the time you're done with your schooling, you'll take all of your board certifications to become a doctor, and you'll get all your certificates and doctorates and everything else you need, and lo and behold, you become a doctor. You made it. You are there. Okay, and lo and behold, he has differentiated. That myoblast cell has differentiated. He is no longer a student in the muscle cell game. He is a muscle cell. And so what we see down here is that he is fully differentiated. Okay? And we can tell that by just by comparing what it used to look like to what it looks like now, right? There are specific things that have been created in the cell body here. And a muscle cell maybe is not a great example, but it works, it's fine. Inside of this cell, it looks like a worm, and what a muscle cell is, it's multinucleated. This is not necessarily anything you need to write down, but it makes this example make a little bit more sense. Muscle cells are multinucleated, meaning they have a bunch of nuclei, and we can see them here in purple. And all of those nuclei share basically one cytoplasm and one cell membrane. Okay? So what you are seeing here in the kind of pink and red banding pattern, those are specific muscle proteins, and we can see those being made here. Once this cell was determined to be a muscle cell, it made this MyoD transcription factor, and that turned on this muscle-specific gene. And then this muscle-specific gene made another transcription factor that turned on these muscle-specific genes here that make specific muscle proteins. Okay? And so that's what you are seeing inside of here. All these pink and red bandings, those banding patterns, are produced by these muscle fibers, these muscle proteins that are created by these two orange and yellow genes. They are what create this stuff, okay? They are what create the raw materials that are put into the muscle cell. And that is how he actually changes it, where the myoblast cell actually changes into an actual cell. So the cell was determined to be a muscle cell first, and then it took the necessary measures to differentiate itself from other generic cells. It got a job. It found a job, and its job is being a muscle. Okay? So this all happened because transcription of all of these gene sets that we see up here, the ones I've drawn all over, transcription of these gene sets had been regulated, which is the part that I had introduced here. Transcriptional regulation of gene expression. We took all these genes and we turned them on. We regulated them. We made them active. And when we made them active, myosin and actin and any other muscle-specific proteins were actually created and put into the cell body. Okay? As you can see, he's definitely changed. He's gotten much bigger. He's got all this cool new stuff inside of him, and it helps him become a muscle. Okay? Cytoplasmic determinants and cell-to-cell signaling also help in cell differentiation. And I had mentioned before that MyoD does ultimately differentiate into a muscle cell, but what happens before that specialization actually occurs? Well, we have to determine what that generic muscle cell is going to be, right? The cell had to go through a process known as determination, right? And so there are certain substances, chemical signals called cytoplasmic determinants that will basically signal a cell to turn into something, okay? So there are maternal substances, substances that are given to you by your mother. Okay, that's why they call them maternal. And those substances are in the early zygote, and they influence early development and sometimes even give certain cells jobs. And they call these things cytoplasmic determinants. These chemical messages, they call them cytoplasmic determinants because they interact with cells. And these cells, once they interact with this chemical signal, will turn into something. They will decide, hey, I'm going to go ahead and be a muscle cell, so they turn into something. So these substances, these cytoplasmic determinants, regulate the expression of genes that ultimately affect the cell's developmental fate. Okay? They help regulate the genes that we had just talked about. They are kind of responsible for that transcriptional regulation. Okay? And so this is a very simplistic example of how this occurs. And up here we see, fertilization occur where the sperm contacts the egg. But inside of the egg we see all of these different chemicals. We see, we'll call it just green triangle chemical. We see orange sphere chemical. Okay? And these are chemical signals. They are also known as cytoplasmic determinants, if we're trying to use the proper terminology. And once fertilization occurs, we get our first mitotic cell division, right? If we want to get to trillions and trillions of cells, we ultimately have to get to many, many cell divisions. But there's always a first one, okay? And what do you notice happens after this first cell division? Well, if you look really closely, you'll notice that there's an asymmetric distribution of the orange sphere chemical and the green triangle chemical. There's more green triangle on the right side, and there's more orange sphere on the left side. And so what that ultimately might do, and I say might because, it's not always a foregone conclusion, and different pieces of chemical information are going to do different things to different cells, but generally speaking, these chemical signals, like the green triangle, may very well enter the nucleus and help to transcribe some of the genes in there, okay? And so obviously green triangle chemical is different from orange sphere chemical, and as such, they may turn on totally different sets of genes. For instance, let's pretend orange sphere chemical goes into the nucleus and will turn on specific genes that ultimately code for a head, for instance, okay? Whereas green triangle chemicals might go into the nucleus and interact with parts of the DNA that code for the tail region of an animal, okay? And so naturally, what we will see is that everything from this point out might code for areas in the posterior or the anterior region up by the head. And everything on this side might code for posterior region down by the tail. So in this way, you have these chemical signals that interact with the nuclei of the cells that they're in to help transcribe those genes and give those cells a specific job. Okay, kind of a neat interaction if you understand how that works. And we'll touch more on this specifically when we discuss some of the stuff that revolves around the fruit flies. Okay. The third phenomenon that we are going to talk about briefly is called induction. Okay, and the example that we see here is induction by nearby cells. Okay, induction by nearby cells can also cause transcriptional changes, and this is something that we're going to see in the development of a creature called a nematode in the next video that I do. But induction is basically, again, chemical signaling. Okay, and a lot of this stuff revolves around chemical signaling. It goes back to that chapter 11 stuff that we talked about that was maybe an overlooked but very important chapter. And the example that's shown here is an early embryonic cell, early embryonic embryo that has 32 cells. That's what I meant to say. And these cells down here that are shaded kind of light blue are undergoing some kind of change, okay? And as a result, they are creating some kind of chemical signal. And we can see that down here. This chemical signal looks like the orange sphere that we saw in the previous one. And that chemical signal is being transmitted to the cells just above it. And so these chemical messages, once they are received by that cell, will elicit some kind of signal transduction pathway that goes to the nucleus, and as a result, will probably turn on a certain set of genes. Now, we don't know what that is by this generic example, but what we do know is that this cell right here, or these cells right here, do have an influence on nearby cells to make them turn into something or give them a job, okay? All these kind of boring beige cells up here are just that. They're boring. They don't have jobs yet. And maybe what's happened is that this kind of light mint green cell has received a signal that says, all right, you have a job now. Okay, you have a job now. And now that they have jobs, they're like, hey, buddies, let's give you some jobs too. And so they give their neighbors a job by way of these chemical signals that you see happening here. Okay. There are also, as it states out to the side here, other environmental factors that influence the developmental information. You could have an external signal. Maybe that's how these cells that are in light blue got their initial signal. Maybe it was some kind of external signal, some environmental signal. And remember where this stuff is happening, right? This stuff is happening in the mother's womb, if you're thinking in terms of animals, right, or human beings. This development of the embryo happens in the mother's womb. And there are plenty of chemical messages being shared between the mother and the early embryo. So maybe it is a chemical message from the mother or from the mother's womb or a hormone or something like that that induces even these cells to all of a sudden have a job. And then when they get a job, they kind of send this information cascade to the other cells to try to give them a job. Okay? So if you've noticed, we have really just scraped the surface of the development between zygote or early embryonic cell like you see here and ultimate like final stage human being, right? But I've at least planted the seed of knowledge here, the fact that chemical signals are vitally important to the determination and ultimately differentiation of cells in the early developing embryo. Okay? And as we get into the next video, so I'm going to take a break here. As we get into the next video goes a little bit deeper into it and talks about how we set up the body plan, how we set up the pattern of what the final organism is ultimately going to look like.